Okay, good morning, everyone. So what I'm gonna focus on is sort of epidemiology and some of the epidemiological factors that might be important in terms of driving disease outbreaks following the invasion of a new vector into an area. And this is gonna be take a sort of a pretty broad approach here, and I'll actually borrow much of sort of the natural history from systems other than ambrosia beetles and their symbionts. The main reason being is that I think many of these other systems in terms of vector biology and disease ecology are far better studied than what we know so far, at least, about this shot hole borer and fusarium here in California. And I'll also be assuming throughout, and you can question me about this later on, but I'm going to be assuming throughout that the impact of this shot hole borer is at least in large part attributable to the fact that it's a vector of a pathogenic bacterium. So again, it's sort of assuming that this fusarium here in California is going to turn out to be found to be pathogenic, and I'm aware that that's not certainly something that's nailed down yet. So I want to start off, though, by talking about different patterns of invasion that might emerge following the establishment of a new vector in an area. And so the first situation would be a situation where a new vector shows up in an area. It doesn't bring with it a new pathogen. It may not even necessarily be a vector of any native pathogens in that area. And so this basically describes what's happened here in California with Asian citrus silk, at least until maybe earlier this year or last year. A second scenario would be a situation where a vector establishes, brings with it a new pathogen. So you're getting sort of novel disease outbreaks in an area where they just didn't occur beforehand. And this seems to describe perhaps what's going on here, again, with the shot hole borer and fusarium, if it turns out to be an efficient vector and the fungus is pathogenic. And then the final scenario would be a situation where you have a new vector arriving, doesn't bring with it a new pathogen, but it is able to transmit efficiently a native pathogen in that area. And this describes this system that I really actually have been working on most recently, and that's glasswing sharpshooter and Xylella fastidiosa. So an invasive insect that's able to transmit a pathogen that by most standards is viewed as sort of native in the area, and is at relatively high prevalence due to the activity of other native sharpshooters, native vectors in the region. I put up these different invasion scenarios in one sense to highlight the fact that it's quite likely that these different scenarios are going to impact have management implications, and even sort of the types of questions we want to start to ask. I think the other potential significance of these different invasion scenarios relates to the potential for disease outbreak and the pace of disease outbreaks. And arguably, you could see reasons to believe that there's differences in the potential or the pace of disease outbreaks among these scenarios. I would assume, all else being equal, that a new vector moving into an area, if it's not, it doesn't have a pathogen to transmit, obviously the potential for disease outbreaks can be really low, at least until the pathogen arrives. The opposite end of the spectrum, again, all else being equal, the highest pace of disease outbreak would likely be a scenario where you have an invasive vector moving into an area where there's already a host of other native vectors, which keep sort of pathogen infection levels or prevalence relatively high. And then you might expect that this novel vector, novel pathogen scenario might be sort of intermediate between those two. But the details of disease outbreaks and the likelihood of disease outbreaks can be affected by a range of different epidemiological factors. And I'm showing some of them here. There's more. Just in the interest of time, I'll focus really just on these four. Again, focusing on systems where we know actually quite a bit about the biology of the vector and the pathogen. And hopefully these will be a little bit informative in terms of our future work in understanding this invasive shot hole borer and fusarium. So the first place to start is obviously talking about the efficiency of the vector. And so an invasive vector moving into an area could have a big impact if it's an efficient vector of the pathogen. And I need to sort of define, perhaps for some people in the room, a really important epidemiological parameter, and that's this value R naught. And you can think of this basically as the disease invasion threshold. And it has a threshold quantity of, at least in the deterministic sense, has a threshold quantity of 1 in order for an outbreak to occur. And so what's being shown here in the figure is the output from a really generic, simple disease model. And so R naught here is plotted on the y-axis, as a function of vector population density. And it's actually, I'm simulating here two different vectors. One, the solid line is for an efficient vector, and then the dashed line here is for one that's basically 1/5 as likely to transmit the pathogen per unit time per insect. And there's two really obvious outputs here, two things you hopefully can see. And the first is that vector abundance contributes to disease outbreaks. So more vectors equals higher potential for disease outbreaks, higher values of R0, well above 1 in some cases. The other take-home point from this is hopefully another obvious one, and that's that the efficiency of the vector also influences the potential for disease outbreak. And there's also management implications here in terms of the feasibility of vector control as a disease management tool. But there's a lot more sort of biology behind the transmission and the efficiency of vectors. And one of the important components of that is natural infectivity. And so what's being shown here in this figure is some data for a leafhopper vector, a sharpshooter, and the proportion of those vectors that are carrying this pathogenic bacterium, Xylella fastidiosa. And the main point is, at least in this situation, the natural infectivity of the vector varies quite a bit over the course of the season. And one of the questions I'm sort of interested in, I'd be really interested to hear from those of you in the room who are a little bit more familiar with the biology of ambrosia beetles and their symbionts, is the extent to which, what might you expect in terms of natural infectivity of these insects? Given the really tight associations between these insects and their symbionts, you might expect that their natural infectivity might be invariably high, quite consistently high, regardless of season or geographic location. At least that's sort of a basic prediction. Another important aspect of transmission efficiency relates to infective dose. And this is the idea that there's some sort of critical inoculum supply that's required in order for disease to manifest in a host. Okay, so a certain number of fungal spores, bacterial cells, viral particles, whatever. And so this is, again, data for Xylella fastidiosa in coffee and citrus. The main point being is that at higher infection inoculum supplies, you have a higher probability that the infection is going to develop into becoming a chronic thing. And so with respect to ambrosia beetles, I think the relevant questions here are not only is there sort of an effective dose with respect to, say, the number of spores that are being carried in the mycangia of an individual beetle, but you might also, an alternative metric might be sort of the number of beetles that are required in a mass attack in order for a chronic infection to develop in a host. So the second thing I want to get into relates to the behavior of the vectors, these invaders, and what it might mean for causing disease outbreaks. And I'll really focus on preference associated with differences in host infection status, whether a host is infected or not. And there's a bunch of examples in the plant pathology literature that vectors orient to hosts based on their infection status, that you might use visual cues or chemical cues or a combination of them. And I think the notable thing is that there's a wide range of outcomes associated with these preferences. And so we see, in some cases, some pathosystems, the vectors show really strong preference for the infected hosts. Other cases, they really strongly discriminate against them and are much more readily going to orient to and feed on a healthy host. And then there's these really interesting examples where there's sort of this mix of behaviors going on where they're initially attracted to these infected hosts, but then after feeding on and realizing it's not such a good host, they'll actually bail off of that, and they can impact transmission and disease spread. And so just to give you a sense for what I'm talking about, here's a couple of different systems that are at sort of different ends of the spectrum here. So on the left is some data we've collected for sharpshooters and their preference based on Pierce's disease status. And so the gist of it is that if you give sharpshooters the choice between a healthy grapevine, and one that's showing these kind of clear-cut water stress disease symptoms, that they pretty regularly orient to and feed on just a healthy one, 2 to 3 times more likely than landing on the infected ones. The opposite on the spectrum would be one of a number of aphid-borne viruses. So green peach aphid and potato leaf rot virus is a case where the infected hosts actually release these volatiles. The aphids use that and preferentially orient to those infected hosts. And so why do we care about these preferences? And so this is at least based on epidemiological theory, there's the potential for these preferences to have really dramatic impacts on disease spread. And so what's being shown here again is output from a pretty generic model. I won't go into the details of the model. But the main thing I want you to focus on is this axis here. And this is basically the vector preference score. And so at this end of it, down near 0, it's describing a situation where the vectors really avoid those symptomatic hosts. The opposite end of the spectrum up here, well above 1, the vectors strongly prefer to feed on those infected hosts. And hopefully you can see that as that value of P increases, you get higher potential for disease outbreaks. So generally, the expectation should be that when vectors prefer these infected hosts, it encourages more and more disease spread. And so with respect to ambrosia beetles, one of the questions that I'd be interested in hearing about, a little more details about, is the extent to which, so the literature suggests that in several of these situations, the beetles are actually attracted to either stress and or infected trees. And I'm curious to the extent to which that's actually really important in terms of maintaining infectivity in these beetles. Is it sort of requirement to have a fairly high prevalence of infectivity in these insects? The next thing that might be important in terms of promoting disease outbreaks following the invasion of a new vector relates to the infection dynamics in a host. And so I need to define two things. First off is the latent period, and this is the time to infectiousness. And so after a host gets infected with the pathogen, how long does it take before it's actually a source of the pathogen for any vector that's feeding on it? A similar, or at least a related issue is the incubation period. And this is defined as the time to symptom development. So how long after infection do you actually start to see any kind of disease symptoms? And both of these things can vary quite a bit among different pathosystems as well as a function of different other variables. One of the important things about both latency and incubation period is that the relative duration can be really important in terms of management of diseases. And sort of from an agricultural perspective, the best case scenario would be a situation where both the latency and the incubation period are both really long and they're sort of more or less equivalent in duration. So infectivity and symptoms are popping up more or less at the same time. Worst case scenario would be a situation where symptoms are manifesting far later than infectivity. So it doesn't take much time for these things to be a source of the pathogen, but we just don't see the disease for a long time. So that gap there, during that gap that I'm showing there in that red box, that's sort of the duration where the host is infected, it's a source of infectious vectors, we just don't know. We don't know that it is. And so this obviously undermines our ability to use things like host removal, roguing of diseased trees in order to limit disease spread. Okay. And to give you a sense for why that might be a problem, here's some data for Wong bean disease and citrus and Asian citrus psyllid. And this is from a greenhouse experiment that some colleagues in Brazil and I conducted where young citrus trees were infected with the Liberibacter pathogen, and then symptoms were scored over time as well as the acquisition efficiency of groups of psyllids on the plants. And the main point here is that symptoms don't pop up until well after 6 months in this scenario. And it turns out in the field, actually, there's evidence that duration actually might be on the order of 2+ years. However, vectors are able to acquire from these infected trees. So the trees are infectious after less than 2 months. Okay? So at that first vector introduction date, we're seeing acquisition already. And so what this is indicating is that there's a really long asymptomatic infectious phase here in this disease. And that's, again, problematic because it limits our ability to use things like roguing to limit disease spread. And so I'll wrap up by talking about issues related to sort of systems where you have really broad host ranges, like seems to be the case in several of these ambrosia beetle systems. And so one of the notable things for generalist pathogens is that the disease dynamics in these systems frequently depends on host community composition. So the abundance and the diversity of different hosts, trees, say, in the community. And the reason is that different species of hosts quite regularly vary in terms of their reservoir confidence. So their ability to support the pathogen. This has important implications for disease dynamics. And so I'll touch on two different dimensions related to this issue of host range and diversity. The first being this phenomenon referred to as the dilution effect. And this is something that's been really well studied in a couple of animal pathogen systems, although I think there's evidence that similar kinds of scenarios might be important in certain plant pathogen systems, where both the vector and the pathogen have really broad host ranges. So in Lyme disease, for example, diverse host communities have lower risk of disease spread, and that's shown here in this figure. And this is some really nice work done by Rick Osfeld out in New York, where they looked at host community composition in these woodlots and related that to the density of infected nymphs, which is their metric of disease risk. And the main thing you'll notice is that out here, when you have these species-rich communities, there's lower risk of disease spread. And that's because in those situations, that high diversity reduces the probability that you're going to have a bunch of these sort of highly competent weedy species. And so in that Lyme disease situation, it's one particular reservoir host, the white-footed mouse, that's really problematic. And white-footed mouse is less dominant in those diverse communities. As I mentioned, certain plant pathogen systems sort of have some of the basic features that might indicate that this diversity might have similar impacts in those disease dynamics. And the last thing I'll talk about relates to differences in host susceptibility to pathogens and what it might mean for disease spread. And the notable thing in agricultural settings is that different species or cultivars of, different species or cultivars of hosts frequently vary in terms of their susceptibility to disease or their disease severity in them. And I want to distinguish between two potential mechanisms that might be explaining that. One is that they could just have differences in terms of their resistance level. And I'm going to define that as sort of a relative reduction in the infection level. So the pathogen doesn't multiply as well in one host compared to another. An alternative mechanism would be that one host is just more or less tolerant than another. Okay, and I'll distinguish tolerance from resistance by basically defining it as a lack of symptoms. Okay, so the hosts are infected; they're just able to cope for one reason or another. They're able to cope with that infection. And there's several reasons to expect that differences in resistance versus tolerance might have differential effects in terms of disease dynamics and the potential for disease spread following the invasion of a new vector and pathogen into an area. And so what's shown here is, again, a little bit more model output, and this is some work that a postdoc in my lab has been doing, trying to evaluate how relative levels of resistance and tolerance might be impacting the potential for disease spread. And I won't go, again, into the details of the model, other than to point out that sort of basically as the colors get lighter here, it's higher values of R naught. And tolerance is increasing along this axis, resistance is increasing sort of backwards along this axis here. So hopefully you can see that the potential for disease spread with high levels of resistance is really super low, okay? Conversely, in the case of tolerance, as you get more and more hosts in the population that are sort of these asymptomatic carriers, so they are infected, they're sources of the pathogen, and they're just not showing any kind of symptoms. That, under most situations, should facilitate disease spread. And that's sort of at least the general prediction of these models. And there's some complicating factors here that I don't have time to get into, but the level of vector preference for discrimination against symptoms could really impact these disease dynamics as well. And so one of the questions related to these ambrosia beetles and their symbionts is the extent to which some long-lived, say, tolerant hosts might be contributing to fungal pathogen spread. And I guess I'll wrap up there, and this is sort of a list of a little bit of a summary of some of the, I think, the topics that hopefully we'll be able to address related to this invasive shot-hole borer and fusarium here in California.